intertwined daughter molecules in the process of decatenation. The dissolution happens first, followed by ligation, followed by decatenation. One of the kind of remarkable things that they were then able to do was to go on and actually now look at the fine details of the time course. To see, is this a smooth transitional process, or is there some sort of event that happens as forks converge one and the other, that they begin to encounter one another, they slow a little bit, they slow a little bit more, they slow a little bit more, and eventually things begin to resolve? Or is it all a smooth linear process? And what they find is, really, if you look at the synthesis and dissolution reaction, it just follows this rather lovely smooth curve. There is no obvious stalling or pausing events. Things happen in this smooth, linear manner. So all the problems that you might imagine of one fork converging and another fork don't seem to be too much of an issue, because they just converge. And eventually things resolve. There is no overt stalling during the termination process. But surely there's going to be some sort of barrier. There must be some sort of issue, because you have these strands converging one upon the other. And so they did again a fine time course, and in this case now they're looking at the fate of the newly synthesized DNA. They're radiolabeling it, remember? They're cutting with a nicking enzyme that cuts the leading strand only. It's a strand-specific enzyme. So the only strand they cut is this red strand here. They'll cut the black strand here, obviously, it's the same sequence, but it's invisible because it's not radiolabeled. The red is the newly synthesized radiolabeled DNA, and it starts out about 300 and it gets longer. Why is it good periodicity? Well, there's sequence preferences within the actual arrays. The arrays themselves are a repeated sequence. You get a little bit of stalling at certain incorporation sites, and that's why you see this sort of ladder appearing. But again, the point is that if we follow the fate of the leading strand, it gets bigger and bigger and bigger. It's smooth. Okay? It's a very smooth, essentially unending transition across the entire array. And it goes across the entire array. Okay? Here we get 12 copies of the array. We see the leading strand going all the way across. And that's weird, because we've got one fork coming this way, and we've got another fork coming this way, yet the leading strand here is going all the way across the array, but presumably the lagging strand should also be coming into from the other side. You'd expect them to collide around about the midpoint here, midpoint being about here. There is no obvious stalling. This is a smooth process. The leading strand just keeps going. Presumably it's encountering lagging strand, but it's kicking it off.
So it means that the strand is
Presumably it means it's a bit like an extreme version of Okazaki fragment processing. We're getting massive level of strand displacement of these newly synthesized bits of the lagging strand. And the final thing that they did was they also did essentially chromatin IP type experiments to look to see what proteins were present when. So they use antibodies against various different components of the replication machinery. So they look at single-strand binding protein. They look at MCM, part of the helicase, CDC45, another part of the helicase, and Pol epsilon, the leading strand DNA polymerase. So if we look at dissolution, so the physical generation of two more or less intact molecules, then that's, as we saw, that happens first. If we then look at ligation, imparting covalent integrity to the daughter strands, it follows this kinetic here. And really beautifully, this shows us the dissociation of RPA, the single-strand binding protein, occurring at essentially the same time as ligation. So the last vestiges of any single-stranded DNA are gone, ligation occurs. And of course RPA, the single-strand binding protein, won't be bound. There is no single-stranded DNA. So it follows the kinetics of ligation just beautifully. But then the kind of amazing thing is that even though all the DNA has now been synthesized and ligated, things like MCM7, CDC45, and even polyepsilon. The polymerase itself, even though synthesis is complete, doesn't leave at the time of ligation. It actually leaves a little bit later. It leaves around about the same time as decatenation. What's it telling us? Well, it's telling us that even after all the replication is done, the machinery is still there on the DNA template. That was entirely unanticipated. Unequivocal.
Dr. Bell,
Hello.
does that kind of go?
Call me Steve.
Okay. So before you said the pCNA ring has the ligase, the fen, and the polymerase on there. Then when the ligase functions, it knocks the other two off.
Is the model, yeah.
So is this kind of showing that's not happening?
Well, we know that the MCM and CC45 are there, and we know from the structural stuff that I showed you before that polyepsilon actually associates directly with the C-terminal domains of MCM. So this could well be entirely independent of pCNA. So yes, there's the pCNA assembly, and that's most important in lagging strand synthesis, where we'll have, well, in the archaea, it's B family, ligase and fen. In eukaryotes, delta, ligase and fen. So the model would be that ligase would ultimately knock delta off, but they don't address delta in this particular experiment. One might expect that it would actually be coming on at this stage. So to summarize the steps, what they seem to be seeing then is we have convergence of one replication fork, another replication fork. We have these pre-catenanes that we've talked about before. This is the interwinding of the DNA behind the fork to compensate for overwinding ahead of the fork. We have CMG trucking along the leading strand template coming in from the right. We have trucking along the leading strand template coming on in from the left. And of course, these are, by definition, opposite strands. Okay. And what seems to happen, at least based on following the leading strand, is that the leading strand here just shoots past the leading strand here. Because one's moving along one strand, one's moving along the other strand. Aha, you see. What about that model you talked about before, where there's two pores in the CMG, one strand coming through one strand coming through the other? Well, this suggests that if those two pores exist, then it's not an obligate requirement that both strands pass through the two pores. But eventually what seems to happen is as this leading strand assembly carries along, it's going to encounter lagging strand Okazaki fragments ahead of it. And what seems to be happening is that this, rather than moving along single-stranded DNA, actually now trucks over and starts moving along the double-stranded DNA, taking the polymerase along with it. Even though the polymerase is no longer polymerizing, it's still attached to the CMG assembly. And maybe delta comes in, does a little bit of processing to clean things up. pCNA will be involved in that. But the point is that MCM, CMG assembly plus epsilon seem to persist in the DNA even after they've done their principal replicative role. They're still there. So how do you get rid of this CMG? We know that you're going to need to recycle MCM back for the next cell cycle, potentially. And there's a really beautiful series of studies that I don't have time to go into any of the detail of, but they're done by Karim Labib and Aga Gambas, two independent studies, back-to-back science papers, showing that at the end of the S phase in eukaryotes, MCM begins to accumulate polyubiquitination, which then targets it for some degree of unloading involving this complex here, P97. It doesn't seem to actually proteolyze the MCM, but actually helps to just rip it off the DNA itself, presumably allowing it to recycle it. So presumably there's something about the nature of MCM when it's bound to this double-stranded DNA that renders it a substrate to this disassembly reaction. And that's distinct from how it's configured on single-stranded DNA when it's still actively unwinding. What that difference is still mysterious at this point. There's another ramification of this idea that double-strand DNA-associated CMG should get stripped off at the end of S phase. Obviously it's important for this termination mechanism, but you'll recall several lectures back I talked about this whole business of dormant origins. MCM getting loaded to all these sites in the chromosome but never actually firing. Well, how the heck do you get rid of that? Same mechanism. It's MCM loaded onto double-stranded DNA. So this will provide a mechanism not only for stripping off MCM that's completed its role, but also getting rid of any stuff that hasn't fired during the S phase, cleaning the chromatin and allowing a fresh reset for the next round of S phase following cell division. And that was the end. Termination was terminated. So, we're going to jump on now. The next topic we're going to deal with, and sadly in this lecture course I'm not going to get to cover everything that I normally cover. Just taking a bit longer for some reason this year. But I want to talk, the next two lectures I want to talk about control of replication. Okay? I'm going to talk today about controlling bacteria. The next lecture I'll talk about it in eukaryotes. The final lecture I'm going to talk about termination. Not termination, I just talked about that. Evolution of the machines. So the final lecture is kind of a soft, fluffy one. But the next two are going to be fairly intensive, so bear with me as we rock through them. So this is the proper lecture 25, control of replication in bacteria. Most of what I'll talk about in the first bolus of this will be E. coli, and I'll talk about a couple of species-specific esoteric aspects if time permits at the end of this lecture. Okay. E. coli, when it's growing slowly, looks vaguely eukaryotic-like in terms of its cell cycle organization. We have initiation of replication. Well, we have cell division. Okay? We divide cells, we're born, and there's a G1 type period of the cell cycle. Okay? We've got a single copy of the chromosome. Replication then initiates at the origin of replication in E. coli, proceeds bidirectionally around the chromosome, giving you daughter cells that are beginning to septate but aren't yet divided, so a sort of quasi-G2-like state prior to division and generation of your G1-like state. Okay? So slow-growing E. coli with a doubling time, about an hour thereabouts. If you're growing it in minimal media, will follow this sort of cell cycle behavior. However, one of the remarkable things about E. coli is you can actually grow it under, well, you can grow it under very rich conditions, and it doubles. Well, I mean, you've grown it in the lab, most of you have, I'm sure, about 20, 25 minutes doubling time. That's fine, but the problem is that the chromosome itself, the kinetics of replicating the chromosome take 40 to 50 minutes. So your cell cycle is actually happening, your division cycle is happening more quickly than the time it takes to replicate the chromosome. How the heck do you cope with that? Well, you become polytroph. Okay. And so what this means is that as you're doubling maybe every 35 minutes, you've got a couple of copies of your chromosome, you initiate replication, you then initiate replication again during the course of the cell cycle, and you actually carry out cell division while chromosomes are replicating from that ongoing cycle. Because you're already oligoploid, diploid in this case, then you're going to divide. These will continue dividing, continue replicating, even as the division process is ongoing, and consequently you'll inherit ready replicating chromosomes in your newborn cells. So the cell division cycle and the DNA replication cycle are to some extent uncoupled. One is ongoing during the process of the other. But how do you actually control replication anyway? It's not just firing constantly all the time. Particularly in slow growing cells, it's very ordered. You fire once and once only for cell cycle. And just as a thought experiment, we know this is the structure of the origin. Okay, we have these low and high affinity DNA boxes. You've seen this slide many times now. We have this filament of DNA A forming. It's held together by ATP. Okay, so it's the ATP bound state allows the propagation of this filament, leading ultimately to melting in the origin region. So how do you control replication? Without looking at the slides, just as a thought experiment. If you're a god, congratulations, you are now God. How are you going, or intelligent designer, let's go for that. How would you design a system to control replication? what stages could you intervene in the process? Sorry, too quiet. Initiation, so yeah, you want to control the system at the first stage. Okay, so that makes sense. You would control it at the earliest possible stage, and that's initiation before you've committed any energy resources to starting replication. But how would you control the initiation itself?
Change the binding. that
Change the binding,
occurs.
yeah.
Mhm.
So one level would be, too many, would be access, okay? One, you could simply prevent the DNA protein accessing the origin itself. How else could you control it?
Limit the amount of ATP.
Yeah, so you could control the nucleotide status perhaps of the DNA. So maybe controlling cellular ATP levels might be a bit draconian because there's so many other processes that depend on it, but it's an essential cofactor for DNA. So controlling the nature of the nucleotide bound to DNA could be extremely important, and it is, as you'll see. In fact, it is the main mechanism. Even simpler than that?
Some other protein factor which binds to that protein.
Sure, so you could control, again, access either by blocking binding to the origin or by binding to DNA and preventing access. Yep. DNA levels itself. You could, in principle, you could actually just change the levels of DNA. Okay. Topology. So we've got access, we've got the concentration of the initiator, we've got ATP, ADP ratios, we've got accessory proteins potentially could be very important. Topology itself, we've got a process that's dependent on unwinding of DNA. If we overwind that DNA, that will counteract the effect of unwinding potentially. So topological control can be important. And the idea of feedback as well. What's this origin doing? Well, it's functioning to set up replication forms. So when would you want to switch off the origin? Well, once you've set up replication forks. You know, if you've actually committed to the point of it working once, then you want to switch it off eventually. And so to do so in some sort of negative feedback loop following the assembly of the overall retrosome would make an awful lot of sense. And you'll see that actually turns out to be the major mechanism of control, which involves some of these other processes. All right, so just going through them one by one. First of all, controlling access of the origin to the initiator protein. E. coli methylates its DNA, and it's this terrible swear word. It's this damn methylase that does it. Okay, and I'm sorry to use such language in civilized country, but damn methylates the 10-6 position of adenosine, adenine, I'm sorry, sticking a methyl group on here. Okay. It does so in the context of this little 4-base sequence, GATC, which of course on the other strand is also GATC. So the A, the base pairs to this T here, is also methylated. So in mature DNA present in the cell, then DAM methylase will identify this site, it will methylate both adenines, and so you will have fully methylated DNA. Of course, if a replication fork passes through this sequence, then the parent strand will have methylated A, but the daughter strand will not. Okay? It will incorporate a standard DAMP that eventually will be methylated back up to form fully methylated. So the idea is that as a fork passes through, and this could be from the very initiation site itself or further down the chromosome, we'll have this brief point where we have this region of hemimethylated DNA. It's a short time window that it exists. It's only about a minute or so before the DAM methylase finds it and methylates it up to fully methylated. Nevertheless, there is a protein called SEC-A which binds to hemimethylated DNA. It specifically binds to hemimethylated form. And it does so by reading this asymmetry within the double-stranded DNA, taking specific contact between the methyl group on the adenine and the backbone of the polypeptide chain. It requires two sites that are close to one another, so it won't just. Oh yeah.
What is the purpose of methylating the adenine?
So part of it is, as you'll see, an aspect of the actual control system. The other thing that's quite useful about this system is during replication, if you make errors in the replication process, then hopefully the DNA polymerase is going to sense those, detect those, and repair those. But sometimes they'll miss the surveillance by the polymerase itself, and you will end up generating a mismatch as a consequence of replication. Then you're in deep doo-doo because you've got two strands of DNA. Well, which one's right? Because they're two different sequences now, they've got a mismatch. Which one's the parental strand and which one's the new strand? How do you tell? And so the way that you tell is you look, the way E. coli tells is it's got a protein called MUT-H, which identifies the methylated strand, says that this is the parental template, this is the one you're going to repair of, and directs the excision of the non-methylated strand in the sequence and allows its repair. So it's an important part of the mismatch repair system in E. coli, is asymmetric detection of methylated DNA. But it also plays a second role, and that's in the actual control of replication via this protein SEC-A. Because it turns out, okay, so that this binds to two sites that have to be adjacent one to the other. It binds with cooperativity, and it can even form these sort of higher order, heavily packed structures. So it seems to be a protein that, in the recognizing hemimethylated DNA, particularly if there's a local high concentration of sites on the chromosome, can actually effectively nucleate the formation of a higher order chromatin structure. Okay. Almost like an epigenetic regulation, I guess, for a bacterium. And the rather sweet thing is that if we look at the REC sequence, which is only a couple hundred nucleotides in length, it actually contains 11 DAM methylation sites, 11 SEC-A binding sites at the origin, a very high density of them. So the idea is that as the origin fires, this will become hemimethylated, and then we're just going to pump in a ton of SEC-A into the origin of replication, and it's going to wrap it up. It's going to sequester it, okay, by binding to it. That's what the SEC stands for, sequestration. So the idea is that SEC-A will bind to the origin of replication, following the initiation of replication, physically preventing the access of any further DNA to it, preventing the reassembly of the replication machinery, at least for a period of a few minutes following the initiation of replication. And we can follow that. So Kirsten Skarsted's lab, who first identified this, she's in Oslo, I think, in Norway, did a really nice chip experiment. It wasn't ChIP-seq, but it was chip using synchronized cells and then interrogating the chip samples with DNA primer pairs at various positions around the chromosome, symmetrically positioned from the origin. So they can do the PCR with these synchronized cell extract, or synchronized cells, and look to see where is the SEC-A as replication proceeds. What we can see is here is the origin of replication, then we've got bidirectional going this way and this way, okay, reading out leftwards and rightwards. SEC-A, following the initiation of replication, is bound to the origin of replication and remains bound for about 15 minutes. Okay. But then gradually it begins to move. Replication proceeds down the chromosome, and we see it transiently associating during replication as well. So it seems to bundle up the origin of replication, remain there for quite some time, gradually releases from there, and then moves out as replication proceeds further down the chromosome. So it certainly seems to be important for bundling the origin, preventing any round of replication, new rounds of replication for at least this period in the cell cycle, but gradually moves out with the rest of the fork as it travels around. And that was supported by microscopy studies as well. Okay, so sequestration is important probably in that it creates this, we call it epigenetic regulation. It modifies the chromatin structure, preventing origin function, presumably by physically competing with DNA. The point I didn't emphasize was that many of the SEC-A binding sites overlap binding sites with DNA. They also are found quite heavily within the unwinding element itself, and again that might help to lock this down, stabilizing the double strand form. Okay, so that's access. What about initiator concentration? Well, that's not really a game that E. coli plays to control its replication cycle because the cellular concentration of DNA is essentially constant across the entire cell cycle. There is some regulation at the level of transcription. So DNA gene itself, the transcription, does get sequestered by SEC8, containing 8 DAM sites within it. And it also autoregulates itself. So there's some cell cycle periodicity to the transcription of the protein or the gene, but the protein levels themselves remain constant. How might you control it then if the protein levels remain constant? Well, this brings us back to the question of access, but also will take us into the nucleotide status of the initiator protein itself. The initiator protein binds to the origin of replication, and that's where it does its primary function. But if you did ChIP-seq analysis, you took antibodies to DNA A, you did ChIP-seq on E. coli, you would find in an asynchronous population of cells, yeah, sure, it's at the origin of replication. It's also at its own promoter, so it's doing an autoregulatory thing. It's at some other binding sites around the chromosome. It's involved in transcription regulation of a number of other genes as well. But you'll also find these loci here, a thing called DAT-A, a thing called DARS2 and DARS1. When we look at the structure of them, there's our 250-ish REC with many binding sites for the DNA protein. DAT-A has a few binding sites for the protein. DARS, again, has binding sites indicated by the triangle. So what are DAT and what are DARS? Well, they're binding sites for DNA, but what do they do? So let's tell you about DAT first. This is a region containing multiple binding sites for DNA that's found not a million miles away from the origin of replication itself. And the thought is that what DAT does is it acts as a sort of high affinity sink for DNA protein. Some people view it as a way of buffering the sort of cellular concentrations of DNA. It's about a kilobase, which in total binds approximately 60 molecules of DNA. And of course, one of the rather sweet things is that the minute you replicate this, you're now binding 120 copies of DNA. So the idea as replication proceeds through this, and it's quite near the origin of replication, you help to titrate away DNA from the origin of replication by providing this kind of high affinity binding to the DNA protein. And indeed, if you take that, as I'll show you in a little while, you do models or levels of replication initiation events. Okay, what about DARS? So DARS now brings us into not only initiator and access, but also this ATP-ADP exchange system. Katayama's lab in Japan, he's really the man. I think it's his review that I gave you to read for this lecture. He's really been the master of DNA for many, really several decades now. And he identified these DARS sites on the basis of a really remarkable property that they have. And this is the idea that the active form of DNA is the ATP-bound form. We know that. That's what allows you to form this filament in the first place, the origin of replication. It's what drives the melting event of the origin of replication. Is the ability of DNA to form these filaments that are mediated by ATP. When you're ADP bound, you're not active. So in order to have active ATP, well, active DNA, you have to have the ATP form. But it's an ATP. That's what it does. It hydrolyzes ATP. And so once you've got DNA ADP, well, how do you deal with it? Well, one is the environmentally unfriendly way of doing it, which is just bash the DNA that's used once, make some fresh stuff. It's going to grab ATP. It's going to open unoccupied active site. It will bind ATP, and ultimately it will hydrolyze down to ADP. Or the other one is the hippie granola Bloomington way of doing it, and that's to do some recycling. And so what we'll do is we'll actually allow ADP to turn over in the active site, be replaced by ATP, and so that way you recycle, literally cycling the protein between nucleotide states. And that's the astonishing thing that this DARS sequence does, is it actually facilitates the exchange of ADP, ATP to ATP bound. Sorry, say that again in English. It facilitates the exchange of ADP for ATP in the active site. ATP replaces ADP in the active site of DNA. again, the idea is the replication of purple trundle through this, you'll suddenly elevate the copy number of this locus, and that will allow you to reactivate any DP bound DNA back to the TP bound form of DNA. It was first shown genetically, and then they went in and did the biochemistry. And so using purified recombinant protein that they loaded up with ADP, they were able to take the DARS site, they narrowed it down to this particular region here, got the incredibly unhelpful name of FK7-7. And if they look at the amount of bound ADP in DNA A by chromatography method, they see when they incubate it with FK7-7, increasing the concentrations of that DNA, they see the loss of ADP from the active site. If they do it at a very low temperature, it doesn't work. And if they make an internal deletion, taking out a binding site within this sequence, again, that doesn't work even at 30 degrees centigrade. So it helps kick out ADP, but does it actually allow binding of ATP? Well, heck, yes, it does. Here we see now that with our FK7-7, as we incubate at 30 degrees centigrade and look at the amount of ATP bound to DNA, we see that increase. It's very similar kinetics to the loss of ATP. So it's actually somehow, mysteriously, we don't know how it works at a molecular level, presumably some swing from the active site, allowing the ATP to fall out. But bear in mind, this is through the act of DNA binding. Okay? This isn't a protein that's coming in and making some allosteric change in the DNA protein. It's actually the nature of DNA's binding to this particular sequence is somehow influencing the active site of the protein itself. And it's really quite remarkable. Yeah.
Oh, sorry. I just wanted to know what's the significance of changing the temperature?
I mean, just that it requires thermal energy to run things. At the genetic level, if you make mutations in the DARS sequences, simply delete DARS1 or DARS2 in wild type cells, well, there really is virtually no effect on growth. This is just wild type background with either no mutation in DARS or the double mutation. I'm looking at, you're all familiar with these sort of spot tests, are you? Just euclideans?
No.
Yeah. No? No. The idea is you plate serial dilution of cells, and the growth of cells tells you how many are surviving, basically. At this level up here, you're essentially saturating the assays. You come down lower here, and you get down to a more sort of individual number of cells that you can count. So for the wild type cells, they're all growing more or less the same. And really, these ones are all saturated, they're almost overgrown. But if you make the same mutations in the context of a temperature-sensitive allele of DNA A, then at 30 degrees, well, everything's growing just about fine. Maybe a little bit less growth of the double deletion in the DNA A TS background. But certainly as you increase the temperature, particularly noticeable at 39 degrees, now none of the concentrations are growing the double mutant in this temperature-sensitive background, whereas the single mutants are a little bit impaired, and the wild-type DARS sequences are growing just fine. We still see, hopefully you can still see growth. Yeah. When you take it up to 42 degrees, well then they're just dead. So basically by slightly weakening DNA A activity via this temperature-sensitive mutation, we then expose a sensitivity to this particular recharging mechanism. Okay. Yeah.
Is there any difference between the DARS1 and the DARS2 sites?
Yes. Will I examine you on them? No. DARS1
Oh.
is slightly
And-
fewer
Okay.
binding sites. It seems to be DARS2 seems to be slightly larger.
Okay.
Okay. What's next? Accessory proteins. The origin of replication contains these multiple sites for DNA, and it contains high affinity sites, low affinity sites, blah, blah, blah. But it also contains binding sites for E. coli. Some people call them chromatin proteins, some people call them nucleoid associated proteins, or NAPs. And the classic NAPs are things like integration host factor, IHF. It's a very, very, very abundant small basic protein, binds to a consensus sequence, and binds actually to the origin of replication. And binding of IHF to this site stimulates replication on this site. Conversely, binding of FIS, another small basic protein that binds, again, semi-sequence specifically, binding of it to this site inhibits replication. How does it do it? Well, the stimulation of IHF, one of the features of the origin is that IHF essentially performs this instruction between these two rather widely spaced DNA binding sites. I mean, I've been banging on for goodness knows how long now about the idea of forming this DNA filament. Well, that's just fine and dandy if your sites are immediately adjacent to one another. You can just bind to here, bind to here, bind to here, bind to here, and then have the ATP helping glue everything together. But here there's a substantial physical space, greater than 40 base pairs of DNA between this site and this site. So how the heck do you form a filament if there's this great gap in between? Well, it's dead easy. You use IHF. IHF produces an almost 180 degree bend in the DNA. As a consequence, you can now allow the DNA bound to this site to interact with the DNA bound to this site. Very, very simple. You're simply physically contorting the DNA, allowing juxtaposition in these otherwise spatially rather distinct sites. 3 dimensions versus 1 dimension. So the idea is that allows you to propagate your filament melting. Yeah.
Is the IHF only binding to like, that specific region and then it's just concentration changing?
Yep.
Okay.
So it binds to that specific site, and it's got a sort of one of these weedy consensus sequences like WATAWWWRX. You know, it's sequence specific and reasonably high affinity, but it's not a classic little binding site. It makes a whole series of contacts in the DNA, and it is literally a 180 degree bend. So the DNA is going to come in, wrap around the dimer. The IHF is a heterodimer, and come out at a 180 degree angle to the far side. So it plays an architectural role. It's called integration host factor, IHF, because it was first identified for its role in the integration of the lambda phage. And again, it turns out that the geometry that it imparts upon the DNA is incredibly important for the synaptic intermediate during the site-specific recombination reaction. But it's a cellular factor, host factor. It's many promoters have it as well. It's well, E. coli promoters. You've got binding site here for a repressor, binding site here for a repressor. IHF comes in, binds in the middle, and allows these things to crosstalk to form higher order structure. Yes.
So the binding of R1 and RSM by DNA is required like at the same time, and that's why that geometry is necessary?
Presumably, yeah. These are quite high affinity sites, and so they're going to get bound, and then they'll get glued together, and that then helps to stabilize the overall.
Okay.
Sorry. And the other thing that I seem to have forgotten the slide for is that IHF levels are high in exponentially growing cells, they're low in stationary phase cells. Conversely, FIS levels are low in exponentially growing cells, high in stationary phase cells. So it's a way of maybe globally controlling replication as a function of the growth of the cells by modulating the levels of these nucleoid-associated proteins. Exponential phase, you have IHF, it stimulates growth, replication rather, and in stationary phase cells, you're no longer growing, you're no longer replicating, you have high levels of FIS, and that just helps to lock down the origin. Okay, getting toward the end of the unimportant points. Topology. As I said, we have a situation where the origin of replication has to unwind. That's what it does. Unwinding will be favored by negative supercoiling. It will be inhibited by positive supercoiling. Okay. And again, the rather cute thing about the organization of the chromosome is that obviously the origin doesn't exist in isolation. It has genes flanking it. It has the MyoC gene to the right in conventional view. It has the GID gene to the left. And it was observed that these genes turn out to have cell cycle dependent regulation. MyoC undergoes a wave of transcription after replication, but not before. Conversely, DNA is on before replication and off afterwards. What do we know about topology? Well, as a machine is moving along DNA, it creates positive supercoils ahead of it, negative supercoils behind it. Consequentially, in this situation, when this gets switched on prior to the initiation of replication, positive supercoils here, but it'll actually negatively supercoil back here, thereby in principle facilitating initiation of replication. Conversely, meiosis is its transcription topology waves drive into the origin region, then it's going to be pushing positive supercoils ahead of it, helping to lock down the origin replication. So even these rather indirect forms of control topology do seem to play a role in governing the overall activity of the origin. So you're getting the message now that there are multiple levels at which the origin is controlled. There's sequestration, there's the ATP-ADP ratio within DNA, there's topology, and there's nucleoid-associated proteins configuring the architecture of the origin itself. But ultimately, and what turns out to be the most important one of all, we're back to Tom Stites' hideously lurid model for the replosome. This is what the origin's about, or this is half of what the origin's about, because it's making two of these, okay? It's building replosome. It's directing the assembly of a replication machinery. And once you've started that process, well, you better not stop it until it's complete. What's the final stage of assembly of a holoenzyme, of a replication holoenzyme? What's the last protein complex that's going to get added? Go through the process. You bring in the helicase. The helicase interacts with the primase. The primase creates a primer, which is then going to be extended by the DNA polymerase. The DNA polymerase requires, for processivity,
The PCNA or
Beta
whatever.
clamp?
the beta clamp, the PCNA sliding clamp. So, yeah, beta clamp. So once you've got your beta clamp onto the DNA, then you've done your business. That's it, your origin is, you can now relax, you can rest. And this again was a really stunning series of studies from Katayama's lab, where they identified this rather derived acronym of RIDA, or RIDA, Regulatory and Activation of DNA A, RIDA. And what they observed was that DNA is the mantra now. DNA is active in the ATP bound form, and what they were able to show, and I'll show you, is that following completion of replication, or following initiation event itself, ATP within DNA is then triggered to hydrolyze to ATP. So that's the off switch, okay? It simply switches itself off. And that this was absolutely dependent on the beta clamp and something else. And through some really beautifully exhaustive biochemistry, proper hardcore biochemistry, they were eventually able to identify this protein called HDA, which was absolutely essential for this process. And it was brutal biochemistry, I'll show you. HDA is kind of an interesting gene. It's conserved in many bacteria to a reasonably high extent. And if you do a blast search, if you take E. coli HDA and you blast E. coli genome, you'll do blast searches, yeah. And then, yeah, you pull back HDA, great, 100% efficiency. What's the next protein you pull out? What's DNA A? So it's the closest sequence relative to DNA A encoded within the E. coli genome. But it's not all of DNA A, it's just my favourite and yours, the AAA plus module with a wee bit of N trans. It doesn't have the DNA P recruitment, it doesn't have the helix to helix, so the classic origin binding site. It's just the ATP is doing itself with this extra little bit of N terminus. And here's the sequence. Its conservation isn't that great, but you can see it's the lineup of HDA from, Chris, I can't remember, what is that, what is it? Is it Salmonella, Haemophilus or something it's called? Cheese. Could be, yeah. Okay, bacteria. I don't do bacteria. But anyway, this thing, whatever that is, a bacterium anyway, E. coli, and then DNA is from the two corresponding organisms. And yeah, there's your AAA plus module. You can spot your Walker A's, your Walker B's, and all that sort of stuff. There's your Walker A, Walker B's down here or something. And there's this little bit shown in green here, which you can see is really nicely conserved between these two different, whatever it is, these two different bacterial species. QLS, LP, E, or F. Look familiar to you? Remind you of anything at all?
It's the PCNA interacting sequence.
So this is the bacterial equivalent of the pip of the teeth. Is one of these conserved. We have a reaction that biochemically requires the beta clamp in HDA. And HDA itself has what looks like, at the level of the sequence, a quite beautiful little beta clamp intact. And this turns out to be the most important of all the systems I've told you so far. This is just an assay for over-replication, miscontrolled replication. If you mutate HDA, you get the highest level of over-replication. If you take out the DAT locus, well, it's kind of blue compared to wild type. SEC-A similarly is actually kind of weak compared to wild type. Take out HDA, you really do get quite significant over-replication. So this is the important one. This is the master regulator. All the other ones do stuff. They're important, they're additive, there are multiple checks and balances in the system. But HDA is of all of them the most individually important. And we have a crystal core. And it looks like AAA plus module. Super. Base domain, lid domain, clamp, binding, teeth at the end terminals. So here's the biochemistry. And this was the sort of experiment you would do to a student, tell a student to do if you really hated that poor student. Because, I mean, this is a vicious damn experiment. What they did was they took wild type E. coli, or E. coli that had been deleted for HDA. And then what Katayama wanted to find out was, well, what's happened to DNA A? Or specifically, what's happened to the nucleotide within DNA A? How do you do that? Well, what you do is you grow your cells with a bucket load of radioactive phosphate, 400 microcuries. That's a savage amount of radiation. And then you tell the poor student to go take this E. coli, which is now screamingly radioactive, and do an IP. So you take an antibody against DNA A, you pull it out, and it's going to come out with whatever nucleotide is bound to its active site. So that's good. I mean, you get a heck of a lot of radiation that you've pulled out, and then you treat it with formic acid, which is itself highly carcinogenic, and then you do some thin layer chromatography with also the buffers for it are fairly nasty as well. And thin layer chromatography, you basically separate out the various different nucleotides. This is your control antiserum, so you have to do a control as well, which is just horrible. So you do it twice, and there's huge amounts of radiation. Hair's falling out, it's nasty. And you run the result out. Well, if you use control antiserum and you're IPing out DNA, well, not DNA, then we have a bunch of radioactive crud that sticks at the origin and never migrates in. If you do it with anti-DNA antiserum, in the wild type cell, we see a spot that corresponds to ATP, and obviously you run marker standards to know this is ATP, and a quite robust spot that corresponds to ATP. If you don't have any HDA present in the cells, then you get far more of ATP and only a weedy little amount of ADP. Okay, so lots of ATP and a weedy amount of ADP. So the percentage is about 70% ATP, 24% in the wild type. Okay. So you've endangered your life to get an exquisitely disgusting looking gel like this. But what it's telling you is that in the absence of HDA, we no longer turn over the ATP-bound form of DNA to the ADP-bound form with as high efficiency. HDA is a clamp interacting. Is that important? Well, then what they went on to do, using purified recombinant protein, purified recombinant beta clamp, purified recombinant HDA, was to incubate HDA with beta subunit, beta clamp, and then run it in gel filtration. gel filtration separates on the basis of size. Stuff coming up at the early stages is big, stuff coming up later is small. The beta subunit of beta clamp on its own comes out in fractions 19, 21, 22, 23, well, these fractions. If we have HDA present, then its bulk of its migration is shifted earlier. In other words, it's forming a complex. So they physically interact. Then what you do, if your students haven't suffered enough, they now have to purify DNA, and trust me, I've purified it, it's not easy protein to purify. And you load it up with ATP, and you start doing more radioactive assays. And you look at ATP hydrolysis by DNA. If you incubate them in the presence of beta clamp and HDA, as you titrate in the HDA, then you see an increase in the hydrolysis of ATP by DNA. Okay. If you mutate that clamp interacting motif, just introducing point mutations, then you either reduce or abrogate that stimulation. So interaction between the clamp and DNA, interaction between the clamp and HDA are required for stimulating the hydrolysis of ATP by DNA. So, it's a AAA plus protein. It triggers ATP hydrolysis by DNA, and interaction with the clamp is needed for these two events. Back to the old structure I showed you many, many lectures ago now, of how typically AAA plus proteins function. Remember, we have a grey proteomer, we have a red proteomer. Oftentimes, these are usually AAAs act as oligomeric assemblies. ATP is the meat in the sandwich between the two different proteomers. Okay. It's bound in the active site of the grey proteomer by your Walker A, Walker B, sensor 1, sensor 2, but the neighbouring proteomer talks via its good old arginine finger, talks into this active site, and that coordination of the arginine finger with the ATP in many instances stimulates the hydrolysis of ATP to ADP by this grey proteomer. So the transacting partner, the red one, stimulates this hydrolysis event. So, in light of all of this, does HDA possibly have an arginine finger? Yes, it does. We look at the consensus sequence, there's clearly an crystal structure, there's clearly an arginine which is the right position within the protein, it's position 168. Don't bother remembering that for the exam, but it has an arginine finger. We set up our assay system with the DNA, ATP, clamp on DNA and HDA. Titrating in HDA, wild type, we saw what I just showed you, hydrolysis of ATP by DNA. If you mutate the arginine finger of ADA, you completely abrogate that activity. Turn it into methionine, turn it into phenylalanine, turn it into whatever you want. Abrogates that hydrolysis. Okay? You do similar experiments, titrating in the beta clamp. Same effect. The arginine finger is absolutely required for hydrolysis of ATP by DNA. The arginine finger of HDA is required for hydrolysis of ATP by DNA. Okay. So, the really cruddy model is we have our filament of DNA. It's melted, the origin of replication, and because I can't be bothered drawing it, it's assembled a replosome. There's a replosome. Okay, that's what a replosome looks like. Slightly less lurid than Stein's example. Final thing that we get when we assemble a replosome, boom, beta clamp comes up. Now we've got DNA filament, we've got replosome, we've got beta clamp bound to DNA. And now that is capable of recruiting HDA. It brings HDA into the immediate vicinity of the DNA filament. And indeed it's so close that the arginine finger of this HDA molecule can talk to this neighbouring DNA molecule. Boom! Hydrolysis. It goes from the ATP bound form to the ADP bound form. That's what we've seen. Okay, they've used model substrates and we've seen exactly that happen. The next bit is my imagination, but we know that AAA pluses are often cooperative in the events that occur. So perhaps hydrolysis of this molecule triggers hydrolysis by this molecule, by this molecule, by this molecule, and so on through the filament. Okay. That seems not unreasonable. How does this filament form? Well, it's partly by protein DNA interactions, that I've said many, many times, but it's stabilized by ATP. Well, ATP is no longer there, so what happens? WHEECH. Okay. Off they go. So fun, I'll do it again. Your filament disassembles. The entire geometry of this thing is going to get screwed up. The origin probably going to walk back down. That's simple. So it's a beautiful example of a feedback loop. You build this entire beautiful Baroque assembly of proteins. The last building block of that, the beta clamate, is the thing that actually triggers disassembly of the stuff that put it there in the first place. It's the dominant mechanism in E. coli. It's been looked at in a number of other bacteria. It seems to be present in them as well. So this is probably one of the main conserved master regulators of replication in bacterial organisms. Does something akin to this happen in archaea and eukaryotes? Kind of, sort of, maybe. I'll tell you about that in a moment. The last few slides, I just want to show a couple of others. I know you're not all complete E. coli aficionados. Remind me, who all are microbiologists here? Who loves E. coli? Who likes bacillus? Who likes Colobacter? No one likes Colobacter. Good choice. Eve Brun leaves the department and that's it. Nobody likes Colobacter anymore. You all just turn your back on it. Okay, bacillus. Let's do bacillus. Bacillus is quite interesting because it does things like sporulation, and that's almost development. Makes bacteria almost interesting. It sporulates in a diploid state, okay? And then it needs to stay. It essentially induces a cell cycle, a post-replicative diploid state arrest, okay, and then sporulates the DNA. You don't want to start trying to make spores and pumping DNA into spores if replication is ongoing. That's going to do all sorts of nasty things. And so this protein here, SIR-A, worked on by Dave Rudner's lab at Jindan Wang's old boss, acts to lock cells at diploid during sporulation. And what you can do is just look at replication foci using DNA-X, component of the clamp. And if you induce SIR-A ectopically from xylose and disco promoter, whatever it is you do with Bacillus subtilis, then what we see are the 0 time point, lovely foci corresponding to replication events taking place, replosome factories being built. If you switch on the SIR-A, then those things just become diffuse. Okay. So it's doing something to just prevent replication altogether. We look at the marker frequency analysis. Without induction, we get a lovely marker, quite a nice marker frequency plot with origin replication. We switch it on, then replication is just stopped. Okay. Not only is it stopped, but it simply isn't initiating at all. And via interaction screens, they were able to show that the action of SIR-A is absolutely dependent on REC. Okay, so you can have a situation where you can have E. coli, Bacillus subtilis, chromosome replication supported by the canonical cellular origin replication REC, or as you've seen, remember the paper from Heath Murray finding the DNA trios and all that stuff? You can actually put a plasmid origin replication in, REN, which isn't dependent on DNA. Okay? If you switch on SIR-A, it kills REC dependent replication. There it is without SIR-A, there it is when you switch it on. So it kills REC dependent replication. If it's REN dependent replication, DNA independent replication, but dependent on all the rest of the cellular machinery, just DNA is the only difference, then it survives just fine. So DNA is the control nexus for SIR-A. And indeed, if one does yeast-2 hybrid, you're all happy with yeast-2 hybrid assays? Yep, good. One can see a physical interaction, a direct interaction between SIR-A and DNA, and the yeast grow quite happily here. And what are we looking at here? Oh yeah, one can carry out depletion experiments to get rid of DNA-A. We're looking now at the SIR-A protein. We see SIR-A when DNA-A is present, forming nice little foci. When we get rid of DNA-A, it disappears. Okay. So DNA-A is physically binding to SIR-A. SIR-A binds to DNA-A and helps to tether it to these specific locations, origins of replication on the chromosome. And indeed, there's even a co-crystal structure now, a bit of DNA bound to SIR-A, so they physically interact. inverse experiment now, inducing SIR-A using fluorescently tagged DNA-A. We see lovely puncta at the origins of replication prior to the induction of DNA-A. Prior to the induction of SIR-A. I mean, really beautiful accuracy. We then switch on SIR-A, and those puncta just completely disappear. It's foci disappear. Obviously, the trivial explanation would be, well, it's just all you've done is you've got rid of all your DNA. Well, that's not the case because Western blotting confirms that DNA fused to GFP is present at equal levels before and after induction. Okay. So what it's doing is it's not getting rid of DNA, it's simply delocalizing it from the origin of replication. So that's a bit all we know about how it functions. It's doing something that interacts with DNA, it's presumably helping to just kick it off the DNA, preventing origin replication. Replication therefore does not initiate, therefore you walk into the non-replicating state. Okay. So Caulobacter, despite Eve's departure, it is actually a really fascinating organism, and it's got this, again, it does development, and it does slightly fancier development than just making a spore. Because what it does is you basically take some swarmer cells, it grows planktonically, it's swimming around, eating food. Eventually it's going to settle down and form stalks, which will attach to a surface. So the swarmer cells don't divide. Stalk cells do begin to divide, and they divide asymmetrically. One remains stuck to the surface. This is like a stem cell almost, I guess. Substratum. And it gives birth to a new swarmer cell that swims off and eventually might go and sit down and then differentiate back into stalk cell elsewhere. Swarmers are non-replicative, stalk cells are replicative. But how do you control that? So they have genetically identified a cell cycle regulator called CTRA, which binds to the origin of replication and prevents replication. Okay, so this is another example of preventing access of the DNA-A protein to the origin of replication. As we see the CTRA binding sites in the boxes here, this box here, this box here, this box here, they're in the immediate vicinity of DNA binding sites. Presumably they're simply sterically improving the ability of DNA to interact with the origin of replication. So CTRA, when it's made and present and stable, it's bound to the origin, stops replication. Great. And as we undergo this differentiation process, well, what we see is the CTRA is degraded during this developmental process. So eventually we end up with a cell that has no CTRA, so it's no longer inhibited at replication, therefore it replicates. It replicates, but then somehow, by a magical process that I'll explain to you, we see this asymmetric distribution of CTRA. The swarmer cell has a buttload of it, and therefore does not replicate. The CTRA slowly disappears within the stalk cell, so replication can initiate again. So we go from a sort of homogeneous distribution of the CTRA in these divisional cells, ultimately to an asymmetric inheritance of CTRA in the two different progeny cells. And that asymmetric inheritance defines their replication fate. How do you achieve this? Well, you use positional cues. The cell has a morphology, it has an asymmetry within it. We have physical cues within the context of the organism itself that tell us information about the nature of that particular cell. What we find is we have proteins that mark the site of the flagellum. We have proteins that mark the site of the stalk. And importantly, these proteins interact with distinct CTRA modulators. The thing that interacts with the stalk, and it's a protein called DIVJ, interacts with a phosphatase. 180 degrees across the cell, the absolute opposite end of the cell, well, we're eventually going to give birth to a little flagellum at this site, so we have this Ple-C protein. That Ple-C protein interacts with something called DIVL, and also so we have a kinase, we have a phosphatase, and the crucial thing that I haven't yet told you is that CTRA itself, and it's written up here, is regulated by phosphorylation. So by asymmetrically positioning a kinase versus a phosphatase, we now have two antagonistic activities at polar opposite ends of these cells. The newborn sperm cell will inherit the kinase. It will maintain phosphorylation of CTRA, and the phosphorylated form of CTRA is the stable form. Therefore, CTRA remains present, replication does not occur. In contrast, the phosphatase counteracts the effect of any pre-existing phosphorylated CTRA, destabilizes the CTRA, so it rapidly clears out. It no longer inhibits replication. Replication can now initiate. There we go. That's the punchline. Phosphorylation stabilizes CTRA, and by differentially distributing phosphatase and kinase, you ultimately dictate entirely different cell fates due to this asymmetric inheritance, positional inheritance, non-genetic. And that, your chaps and chapesses, is it. Quick run through bacterial replication initiation control. Phosphorylation, as you discovered, is a horrifying, disgusting, repellently complex importance of eukaryotes. You shall all suffer together through eukaryotic replication control. Brace yourself. Have a marble.
Yeah, I was like, wait, this makes sense.
Yep.
Oh, yeah.
Really know it's more
Put it on a plate.
Yeah, makes sense. Put it on the plate. We look at it on the road.
I'll text you later.